Welcome back to Data in Biotech. I'm Ross Katz, Principal and Data Science Lead at Cordine. Here's a paradox sitting at the center of AI in biology. Ask almost any computational biologist what they need to build better models, and you get the same answer: more data. Yet, as an industry, biotech barely spends anything on generating data. My guest today puts it about as bluntly as it can be put. Everyone thinks AI is going to transform biotech, but no one is willing to pay for the biological data that AI needs. John Androsavich runs Ginkgo Datapoints, a company built on the bet that data, not models, is what's holding the field back. He came up as an RNA scientist, spent years on the pharma side deciding which technologies to buy, and now sells the raw material everyone says they want and few are willing to fund. We get into why that gap exists, what it's costing the field, and what Ginkgo is doing about it. From ADME testing at a tenth of the going rate to an autonomous lab where GPT-5 designed its own experiments. Let's get into it. John Androsavich, welcome to Data in Biotech.
Hi, Ross. Great to be here.
Well, just to get us started, would you mind? We're talking about Ginkgo Datapoints today, and so you launched in late 2024, and it's been described as defining the category of being a bio AI data provider that's generating biological data specifically for AI model training rather than as a byproduct of a drug design program. I'm interested in, from your perspective and from the perspective of Ginkgo Bioworks as an organization, like what led to the launch of this program? What were you seeing both internally and externally that made you think this was the right thing to be doing?
Yeah, it's a great question. And I think Datapoints really was able to emerge at an important time in the AI story as it's been applied to biology. And so this is 2024, just to kind of set the time frame. AlphaFold was going to win the Nobel Prize in October. This would be associated with Demis and John Jumper, David Baker. So a really big boost for the AI bio space. We started thinking about Datapoints probably a few months before that, the summer of 2024. And really, I joined Ginkgo in January that year. And looking around and seeing the resources that were available here, we have 250,000 square feet of space in Seaport, Boston, that's chock full of lab automation and advanced analytical equipment. It's a really superb facility. Most of this has historically been applied to what we now call our solutions business. That has a slightly higher barrier to entry. You're doing strain production. Maybe you're making antibody. There's like a physical outcome that comes out of that. But we're really thinking about what are ways that folks could take a smaller bite out of this kind of lab facility. And data, number one, seemed to be obvious, but also understanding that if AI were to become more performant in biology, it would need domain-specific data. And that data would not only need to be scaled, but it would need to also maintain a very high quality control. This is only achievable through automation and not through pipetting. Now, there are other ways to generate data, pooled experiments, kind of these surrogate outcomes, et cetera. But what we wanted to do with Datapoints was enable the industry to buy the same data sets that they buy now at a lower scale, but now at a much higher volume. And that's only enabled through the automation, all the great, analytical instrumentation that's available at Ginkgo today. Now, what's interesting, Ross, is that the market reception to this was a little soft in that there were early adopters that were happy to serve. However, at that time, there was, and this is only 2 years ago, it's amazing how far the field has advanced. But at that time, there were a number of companies that were really interested in working with us. They could see that they could benefit from us, and that was mutual. However, they were a little scared off by the AI. They didn't see themselves as an AI company, and so maybe that wasn't for us, and they just wanted to maybe be a little bit reluctant to jump in there. So we actually had to adapt our marketing a bit to soften that position. And I think that we've actually seen now the tide turn, and the AI space is much more ubiquitous. It's becoming even more so. People aren't turned off by it. And we serve a number of customers, some that want the large AI training data, as well as others that are using maybe AI models on their side, but they need validation. They kind of need lab in the loop and it's lower scale. But this now offering has really turned on and grown with the adoption of AI within biology.
I'm really interested in looking under the hood of the work you're doing from an automation perspective. But before we do, you know, what are the types of organizations or the types of the types of programs that tend to benefit most from the data that you're able to provide?
Great question. There's we work with everybody across the spectrum. That's one of the things I love about this job. So we work with small biotech, large pharma companies, small tech bio that are really more technically inclined, less biologically, and they're usually kind of the AI developers in the space. But then also you have a really strong entrant, especially recently, of large tech firms getting into the biology space too. So we help them across the board. We typically work in a few different areas. We work on those pillars that are really key right now for biotech AI training. That is in the protein space. We're working on antibodies and antibody developability. This is really characterizing an antibody, and does it have drug-like properties? Does it go from just being a biological molecule to potential therapeutic? We also do the same thing in the small molecule space. This is chemicals. Is it just a chemical or could it be a drug? And this is really measurement of those characteristics. And then we're also looking at the cell level and understanding how are cells responding to different perturbations, whether that be chemical perturbations or genetic perturbations, and really trying to help train models for what are now called virtual cells.
Interesting. And when we were talking earlier, you talked about this paradox in the ecosystem where everyone believes that AI is going to transform drug discovery, but everyone says that the major bottleneck to better AI is more data. Yet the investment that people are willing to make in data generation in biology is still relatively limited. So would you mind just fleshing that out a little bit? Why does this paradox exist?
Yeah, it's fascinating. Every time you see a survey and you ask computational biologists, AI researchers in the biospace, what do they need to really improve their models and get better? Almost uniformly, you see more data, right? But let's put it bluntly, and I think this is, I'm going to state it a certain way for the sake of illustration. Everyone thinks AI is going to transform biotech, but no one is willing to pay for the biological data that AI needs. That's the paradox, right? And I'm going to repeat that last bit. No one is willing to pay for the biological data that AI needs. So that's a bit of an exaggeration, obviously. In recent news, Chai Discovery, a biotech modeling company, they just raised $400 million, right? I suspect a good portion of that will actually go to new data. Even bigger, Isomorphic a couple of months ago raised $2.1 billion a month. 2.1 billion. And that's, maybe some of that would go to data. Other actually would go to maybe clinical trials. They do want to develop drugs themselves, unlike Chai. So let's say that there are important investments being made in this space, and I'm extremely excited about those investments. But I think those are exceptional examples, and it's worth putting the entire industry into perspective. So let's compare biotech to tech. Right? Just to label training data that already exists on the internet, but not to label it for LLMs, 3 vendors alone pull in something like $3 to $4 billion in annual revenue. And that's growing fast, right? Meta paid about $14 billion for a minority stake in one of those, Scale AI. And so that one check actually represents more than a full year of all AI drug discovery venture funding and several times the size of the entire single cell data market. So what you get is, even before you account for hundreds of billions of dollars of capital expenditures that tech is pouring into data centers and AI compute, Ross, by comparison, biotech looks like it's business as usual, happy to just flirt with AI.
Yeah, no, that's it. It makes a lot of sense. And it's also sort of counterintuitive because a lot of the conversation in biotech seems to be dominated by the idea that these computational tools are going to lead to, less expensive drugs coming to market, or it's not going to be $10 billion to bring a drug to market. It's going to be something closer to one, and it's not going to be a decade to bring a drug to market. It's going to be a much shorter period of time. So can we talk a little bit about the reasons for this underinvestment? So one of the reasons that you mentioned previously was this idea that biological data is more expensive to generate than scraping the internet. But at Datapoints, you have this offering of ADME 1 your product that offers, $199 per compound. So I'm interested in, how you think the automation that you're able to bring to bear is able to change the cost structure and what that means in terms of the level of investment that companies can make and what they're able to see from their investments in data from in biotech.
Yeah, thanks for mentioning ADME 1. We're really proud of that product. It's about, just to put it in context, about 1/10 the price of where you can get those services anywhere in the world. Anywhere in the world. And there's some really extra features that are special that go along with that. And that's not even just counting the price alone. That was enabled through automation, but also through smart partnerships. So I'll give a call out to Inductive Bio and Tangible Scientific that are our partners in ADME 1. And if there's interest in that, certainly we've seen a really strong demand for that because it is such a stellar price point. And overall, though, this is classic Jevons paradox territory. Okay, so the more efficient a resource becomes, the more money is spent on it because consumption goes up. And it's not just that biological data is expensive, it's that creating new data in general is expensive and also takes time. And so what we wanted to do with Datapoints is that we wanted to scale data production and we wanted to capture those efficiencies through that scale and through that automation and pass those savings down to the customer with the idea that the customers would effectively buy more. They would use their dollars that would be much more powerful. They'd be able to train more performant models, and this would start that flywheel. But the other part of it is that actually turnaround times are important too. So we really stress turnaround times. We make our business as seamless as possible so that you can get the data that you need, whether that be for a large training run or it needs to be for like a validation lab in the loop type setting. Automation, of course, is the linchpin behind all of this. We like to think about automation not as a human replacement, but as a multiplier on every FTE that we have. And so humans are still a really important factor into this, and I can't understate that or overstate it. I'd argue that they're actually the most important factor. But each one of our scientists can actually accomplish much more than that they would have been able to do alone with a single pipette now that they have automation. And that has a huge impact on productivity, has a huge impact on direct costs. And of course, we hope that has a huge impact on the types of data we can generate at a price point and with a certain time turnaround time that would allow our customers to really feel like they are enabled and empowered to train better models.
Yeah, it's interesting to hear you. I mean, we're sort of in AI data land, so I guess you can't record a podcast in AI data land without mentioning Jevons paradox. But but I'm interested in how you evaluate the validity of that paradox inside of it, like inside of biotech. Like, what would you need to see in the marketplace, or what would you want to see in the marketplace that would tell you that, as the price goes down, that the demand, will increase even beyond what you would expect?
Oh, we're already seeing it. We're already seeing it in the way that folks are using this. And as I mentioned, there's a range of customers that we work with, and a lot of folks are using ADME-based prediction models, and then they're fine-tuning those ADME models using ADME 1 for their particular chemical series, right? What we're allowing with this price point is allowing them to do that earlier and sooner so that they don't need to triage their molecules so quickly. A lot of times we conduct a drug discovery campaign, and the idea is that we have this funnel, and very quickly we want to narrow down to just a handful of molecules. In order to get there in an efficient manner, the conventional way is that you just make triaging decisions. And the idea of ADME, it's funny because there's different tiers. You always talk about tier 1 ADME. Now, I'm, mostly from the RNA space. I've done a lot of oligonucleotide discovery, a lot of RNA, LNP type discovery. Small molecules, I'd have to go back to grad school when I was doing chemical biology work to really familiarize myself with the space. But, now that I'm back in it with Datapoints, I've done so. And it's given me an opportunity to kind of look at ADME with a fresh set of eyes and a different perspective. And when I first came in here, I had the naive perspective that tier 1 ADME was something that everybody agreed were the most important assays. What I learned is actually it's just what your budget allows. And so tier 1 ADME is 1 or 2 assays, or maybe 3 or 4, depending on how much of a budget you have. Either way, the idea is that you eventually find a number of compounds at the end and they get a full workup. What we're seeing now and what we're enabling is everyone to be able to make their measurements of the full tier 1 panel that fits in everyone's budget early on and in more molecules. And I think this is really important because it allows you to make more effective decisions on your drug discovery program. And so that's kind of like a one-time consequence or a one-time payoff. But even broader than that, it allows you to generate data that effectively could be used in model training. And so one of the things that's missing if you triage everything first is you're missing a lot of negative data. And so by allowing folks to actually buy more early on, it does give you that ability to have the negative data that can be used for downstream models. So I think it's paying off, and we're certainly seeing this in the way that we're engaging customers and that they're coming to us earlier and asking for larger orders.
Yeah, that's interesting. What I'm hearing is that, basically the availability of the lower price ADME, ADME assays gives you the ability to de-risk earlier in the development of the drug, and then also because you're able to get a higher volume of data and these models that people are using for predicting things like ADME are so data hungry. By its nature, sort of the drug discovery program writ large across organizations becomes one that relies on that data sooner and sooner, which sort of logically leads to the conclusion that you need to acquire this data. You need to acquire this data as early as possible, which the price point allows.
Absolutely.
Am I thinking about that right?
Yep, that's exactly right.
Yeah. So, another thing that I've heard you talk about related to underinvestment in biological data is this idea that we don't yet know the scaling laws for biological AI. How much more data leads to better performance, across tasks, or in the various tasks that we have in the biotech data space. And at what point returns start to diminish. And, so this uncertainty causes hesitation on the part of potential buyers for biological data. So I'm interested in how you advise biotech organizations to think differently about data acquisition and, start to try to characterize what scale of data they need to generate in order to accomplish their goals.
I think the debate is still out on this. You can look at recent papers, and there's some really good ones. But it depends on the domain that we're in, and certainly protein language models, they seem to scale better, maybe than some other ones, similar to large language models. Two examples I want to, I think, illustrate, the way that I talk to customers and the way that we think about data coming in. So the first one is actually from Microsoft Research, and their academic partners. They just released a paper this month, and what it shows is that single cell foundation models, they don't follow scaling laws. So I think we're learning a lot more about what doesn't work than what does work. So it's still progress, maybe just slower progress, but this one's really interesting because we always think about single cell, or the field thinks about single cell as being maybe the epitome of good biological data that you can train on, because it's relatively easy to get large amounts of data. We're talking about 100 million cells, lots of cells. Each one has multiple data points in it. You're measuring the transcriptome or the RNA expression in each of these cells. And it also allows you to have heterogeneity. And so, that's a really powerful thing in biology, because if we think about a tissue, that tissue, depending on the tissue, could have 6 or 20 or even more different cell types. And so having single cell methods has been revolutionary for biology. And I don't want to dismiss that. It's absolutely true. However, for modeling, it doesn't seem to be as performant as what you would expect. And so what this Microsoft Research paper showed is that you can take 20 million cells for training, but your learning saturation actually plateaus around 200,000 to 2 million cells. That's a 1 to 10%. And then you don't get any benefit beyond that. I think this is a really surprising outcome for many in the field, maybe a little disappointing. But we'll talk about other methods that actually data points that we've been using that we really think are more exciting and probably more information rich that could help get over this saturation gap. But I think that's one example where maybe the biological scaling didn't allow for the AI performance that we would expect. Another one is in our own hands here at Datapoints, and that is, I mentioned we work on antibody developability. Now, antibody developability, through our best efforts, is still relatively expensive. You have to make the DNA, you have to make the protein, and then you start to measure these characteristics. We are actually making gains and improving the efficiencies of that, and I think there's some tricks, and we hope to announce some stuff very soon. But for now, we're talking about hundreds of dollars per antibody, right? So we test these, and we have one example where we have 250 antibodies, and we have some basic predictive models in that. You can learn on 250 antibodies. But we also have, one that is 10 times higher of 2,500 antibodies. That allowed us to use some more advanced models like protein language models. We were starting to get into that big data paradigm. But we compared the performance of each of these models, one trained on 250, the other one on 2,500. What we found is that the improvement with 2,500 was not 10x better than 250. It was better, but not 10x better. I think this was a little bit surprising. However, we went back and we looked, and the catch is that the 2,500 antibody data set was actually less diverse than the 250. And this really points to an important decision-making step when you are designing data for training. And that is that it's not just the abundance of data, single cell being an example, antibody developability being another example. It's not just the quantity, it's the diversity of that data, and it's the types of the data that really matter.
Another thing you've talked about related to, like biotech companies' orientation toward data generation is that, every company that has a foundation model sort of believes their model is already the best, and so there's not an incentive from an economic perspective to invest in improving it further with additional data. And there's this is partially because you've got a strategic orientation where companies are competing on sort of the narrative underlying their model and the pedigree of the researchers that have released the model rather than the model performance itself. So I'm interested in your perspective on this dynamic and, what it looks like in the field.
Yeah, I have an insider perspective on this, being on both sides of the table, that is pharma buy side, doing search and evaluation, and also now on the biotech sell side. And, what's funny is I saw a post from Alex, the CEO of InSilico, on LinkedIn, a few weeks back. Now, if you follow Alex, he often offers some really hot takes. And so very interesting guy to follow. But in this case, what he suggested actually makes a lot of sense. And that was, how about if we could create an AI agentic method for doing biopharma BD, right? It's such a wonderful techno-utilitarian view. But, the problem is that biopharma partnering is a marketplace. And, like the economist Richard Thaler pointed out, is that marketplaces are not always rational. It's classic behavioral economics. And so there are so many different aspects at play in any pharma partnering decision that goes well beyond, what is the best model. And we'll talk about how do you even evaluate what is the best model. But, it means that you have to start thinking about, does it fit my strategy? Who is the internal champion? What are their motives? Is there endorsement from the C-suite? Are there pre-existing relationships at play? How might geopolitical issues play into this? That also has a lot to do with it. And so often it's more than just having good data or good models. And it's that story component that really is fundamental if you have a leg up from your competition because of your pedigree, because of your investors, because you have these big names. I mean, this is part of the idea that when VCs are investing, they invest in the jockey and not the horse. And so I think that having a story is really important. Now, the question is, how do search and evaluation folks actually make a decision on who they're going to partner with if they truly want to find the best technical solution? And we could talk about some concepts for that. What are the best frameworks? But in general, I would say that you're limited as of today, and you're often extrapolating on potential, right? So that's what everybody's doing. And, that can really be a situation for these model developers where they might be talking to their board, thinking about the best way to deploy their cash. And they're not sure unless they're in a head-to-head competition with somebody, how do they demonstrate the value of doing additional data generation and having better performance? Certainly a competition would help there, but also it could hurt. And I think there's some risks there. So oftentimes we get trapped in this narrative. I think both sides get trapped in this narrative of thinking that let's do a BD deal, let's find our pharma partners first, and then we'll invest more into our models if we need to. And I think that in general is a little bit of a break on what would be some additional investments into this field if we did have a more empirical way to evaluate the winners and losers.
Yeah. Well, since you brought it up, what would that empirical way to evaluate the winners and losers look like in your opinion?
It's a great question, and one that we thought about a lot from Ginkgo's side. I think that we could play a big role in this. However, we don't necessarily offer the incentives that some of these premier bio AI companies would need in order to participate, right? So I would say the number one condition that you need here is actually to have a large pharma do an RFP, a request for proposal, and they would actually seek out and invite these bio AI modelers to participate in an internal competition. What that could look like is you have maybe easy, medium, hard targets. If a biotech company wants to participate in only the easy, because that's where they think they can win or that's best aligned with their model, so be it. That's what they can choose to enter in. Regardless, they would submit their submissions. This would be all in silico-based designs. Maybe it's for an antibody that binds to a GPCR, something like that, receptor. And they submit that sequence, and then you would have the pharma company or a third party to actually evaluate the outcomes of that in the wet lab. Then you would be able to compare across these various submissions who are the winners for that specific task. And if the company is smart, they will actually put meaningful tasks on there, something that is relevant for their portfolio. So then the winners they can choose and partner on. I recently heard J&J, I believe, is doing something like this now. So I think this is starting to take off. We'll see where it goes. I do think that there's a benefit in having that empirical evaluation and being able to pick the best one in the bake-off, I think is good for everybody and good for the field. Again, does that really fit, though, that like kind of irrational marketplace? Will you have really adoption on this, or are you going to lean more on narrative if you think that gives you a leg up? We'll see.
Yeah, I mean, just hearing you talk about it, I can think about the strategic considerations from both sides. And, from like a modeling company perspective, you're trying to only incorporate the data that you need in order to get that market validation that you can, and, get to the level of partnering with the organization so that you can, solve real drug discovery problems that lead to revenue for the model. And then you can tell yourself a story about how you're going to, program after program, like, climb your way up and broaden the ability of the model. But the idea of purchasing enough data to feed a foundation model that's going to characterize sort of the breadth and diversity of drug space, regardless of the modality that you're using, it sort of seems out of reach. And then from the pharma perspective, you're willing to try anything that might get you to a high quality validated target that you can bring to market faster. But the idea of sort of investing the money without the validation up front is too much to get over. So there's sort of like the I can see why that deadlock kind of arises. Do you think I'm thinking about that right?
Yeah, absolutely. And I'm glad you mentioned money too, because that's one dynamic I hadn't considered in this. And that is, if you really want the best predictions from these best modelers, it is not free. Inference is expensive. And so just coming up with the predictions is really kind of a task that you have to figure out. And you don't want to be in a situation where companies are not putting forward their best prediction because maybe they're running multiples of these. If they have to do this for every single pharma company, it becomes prohibitively expensive, and then maybe they're not putting their best foot forward. So I think there's a lot of details to work out in this type of framework, but ultimately I do think that the field will evolve and eventually get to something that is more level setting. And, we're all scientists at the end of the day, and it will be more data oriented than what it currently is now, just through extrapolation.
Yeah, and this sort of puts into relief the kind of rational defection that you've described about how there are players in the space that are more than happy to just let other players invest in the generation of data and the development of models, and wait to invest until the inexpensive solution is available to them. Is there a case that you would make against that kind of logic, sort of sitting on the sidelines?
This isn't new to our industry, the biopharma industry. It's been happening for some time now. You can actually look at over half the drugs that are approved today. They don't originate in the pharma companies that sell them. Okay, they come from biotech, and that's now the working model. So VCs fund early innovation in biotech. Pharma buys it up later. And that's just the way it works. Now, that's not to say that pharma doesn't do good research themselves. They absolutely do. But their risks are a little bit more less, and they tend to be more conservative. And that's buffered by the fact that they can always come in and buy it later, right? Now, I think AI is actually accelerating this trend and also shifting it to some extent. I don't think it's necessarily a freeloader problem or anything like that. I think it's more of a paralysis of choice problem, where everybody across the industry, including pharma, as well as smaller modelers, is that in a field that moves so quickly, where can you go and invest your dollars that would be able to compete with your beat your competitors? So it compounds, though, I think, in that there's statements made by leaders in this space that they don't need any new data, right? It's all about model architecture. Now, that statement was made a couple years back, and it's likely been walked back today. In fact, I heard that the individual that made that statement, actually, they're building their own labs, and so maybe they're coming around to it. But I think that type of statement, that kind of thought, whether true or false, I don't know, and it's probably somewhere in between. In some cases, it is actually right. But I think it scares people into looking foolish, right? They don't want to look foolish. They are concerned about taking the expensive approach, buying the data, and then being proven by someone wrong that, if with better AI researchers, they could have figured it out with the existing data today. And I do think that creates this second or third or fourth, kind of take on it as you're making these investments. And that just slows things down. And the longer it slows down, then you think you're even further behind, then you don't make the investment again. So it is really compounding in that way. So I do think that this is a concern in the space, a problem in the space. I'm interested to see, Ross, how this actually evolves now that the large frontier hyperscalers are getting into this space, right? Anthropic announced that they're getting into drug discovery. Now, there was a time when IBM was also in drug discovery, and clearly they didn't make any drugs. And so who knows how real this is? But is it that you now get less VC funding into these smaller biotechs that are actually more domain specific because the VCs are concerned that the frontier model companies are just going to overtake the whole industry, right? And so that's a real thing. And I think it's a similar problem to what we're discussing here.
Yeah, that makes a lot of sense. It is impossible to say how successful these non-biotech companies can expect to be in the biotech space. The biotech industry also has a long history of people who understand data science and machine learning and statistics and software engineering coming in and thinking that because they've solved complex problems in other domains that they can necessarily solve the most complex models in the biotech domain. So, I think naming IBM's previous drug discovery efforts is an appropriate grain of salt for all of us to take along with this evolution. I want to talk about some of the initiatives that Datapoints has in place and sort of how they relate to the way you're positioning yourself in the ecosystem. So, you've launched this. Can you just introduce us to the Virtual Cell Pharmacology Initiative and, what it's doing and why you decided to launch it?
Yeah, so the Virtual Cell Pharmacology Initiative is really an attempt to get the industry to recognize and use a new method for transcriptomic measurements. This goes back to the single cell data. There are approaches to measuring transcriptomics that are more high fidelity, more high signal to noise than single cell alone. One of those approaches that we use is called drug-seq. Now, drug is actually a pretty contrived acronym. I won't even explain what it is, but actually I think it's on the nose because you can use this method to actually assess the effects of a chemical perturbation. That is, you add a compound, a small molecule compound or chemical or drug onto a cell, and you are able to measure the transcriptomic response, the change in RNA gene expression that happens as a result of that drug. So drug is useful, but you can use this in many different applications. So your perturbation could be a CRISPR perturbation or a genetic perturbation where you knock out a gene and you see what the response is. So it is effectively a very broadly applicable technique. And we do this in 384 microwell plates. And each microwell plate has its own unique perturbation, either drug or CRISPR gene knockout, or you can even do it in combination. You can add biologics to this. Any perturbation you can think of, you can do it. And that's the advantage of doing this on an arrayed format. So that is, instead of doing a single cell experiment that is all pooled, it's all in one tube, and then you use the single cell sorting to really see what the outcome is on each individual cell. These have a literal physical plastic between each experiment, if you will, on that 384 well plate. So each well is an experiment. Now, in each of these wells, we're able to measure 10,000 genes on average. Those 10,000 genes we measure with a lot of reproducibility, and they have very high signal to noise. And we do that on the order of $10 per well. So it is a very efficient technique. And for a full 384 well plate, you're able to test a lot of different conditions and generate a lot of data for less than $5,000. And I think that's a really important technique. It was not discovered by Ginkgo. We have, I would say, perfected it. I don't know if we've reached that end stage. It gets better all the time, actually. But certainly we've been able to scale it. And it was originally invented in 2018 by Novartis. And it's been under the radar. And I think it's mostly been under the radar is because the field, due to influence from a lot of influential institutions such as the NIH, has really focused solely and singularly on single cell techniques. And so I think that, single cell, again, really revolutionary way to measure biology, the heterogeneity of biology. It is alluring because it has such a high amount of data output. But I would actually say that it is, high in calories, low in nutrition. It's kind of like the junk food, if you will, of transcriptomics. While with DrugSeq, we're actually able to have much higher fidelity measurements. And sometimes we can get more than 10x the number of genes. The problem with single cell is you get a lot of dropouts. And so you create this, what's called a sparse matrix, and most of what you're measuring are zeros. Okay, so huge amounts of data, but you have a lot of these dropouts where you're measuring the genes expressed from each one of these cells. And even if it's a highly expressed gene, sometimes it drops out. And of course, the correlation between expression and dropout is very significant. So you often have more dropout on more medium, lowly expressed genes, which are often the most sensitive to these types of perturbations. Now, with DrugSeq, we do make some compromises in that we don't measure every single gene that you could with bulk RNA-seq. That is more on the order of $300 to $400 a sample. Remember, we're at $10 a sample, right? So there are some limitations to that, but we still get 10,000 gene measurements on each one of these samples. So it's a lot of data. We think it's sufficient data to actually train models on. And the idea is with Virtual Cell Pharmacology Initiative is that if we're able to not only tell people that this is the best method, but actually demonstrate it and get the data into their hands, they will start to adopt it more and see the advantages of it. And I don't think it needs to be an or. I actually think an and is really useful. We've had some early adopters of the VCPI data that have combined it with single cell models, and they're seeing actually improved performance across the board. So I think it could be complementary as well, which makes a lot of sense. And so I'm really excited about this initiative. There's also a special aspect of it where investigators, scientists, hobbyists, whoever they might be. Now, the hobbyists may not have access to these types of things, but the invitation is still open that you can send us your own chemical compounds and we will screen those for free. And so you can send us, we're doing 100,000 of these compounds. And so you can send us 10 or 100 or 1,000, whatever it might be, and we will screen those for free and we will put them on the platform and everybody can kind of benefit from this data being out there. So I think it's really unique in that way. It's taking a different approach to transcriptomics, which are really kind of the bedrock of virtual cells. It's offering an orthogonal measurement there, but also we're making it kind of fun in that it is open source. It is all, MIT license. You can use it to train models, you can use it for commercial purposes, and you can actively participate by actually submitting your own compounds.
Yeah, I mean, there's so much here. I want to, there are so many questions that I want to ask, but as you're talking, what I'm hearing is sort of the DrugSeq at $10 per well is sort of, what you all view as a Pareto optimal data point that is both highly informative and, it's highly informative, it's highly reproducible, and it's very cost efficient. So you're able, sort of the argument to the ecosystem at large is here, look at this data. It's very informative, train some models on it and see how informative it is. And then that builds some momentum behind, acquiring more of this data and kind of starting that flywheel. Am I thinking about that right from the data points perspective?
That's exactly right. And, one of the challenges with DrugSeq, I mentioned, that there's, a focus on single cell that maybe is causing folks to overlook DrugSeq. I think the other thing, too, is that it was started in pharma companies, and it really is most effectively implemented within companies that have large-scaled automation, right? Now, pharma companies are not going to open their doors to anybody who wants to do DrugSeq and let them in. But Ginkgo Datapoints has. And so I think for the first time, we're unlocking this technology. We are really democratizing it and allowing people to take advantage of it. And part of it is just awareness. And we're really hopeful that once people not only get to understand the technology, but they see it and they use it, is that it gets adopted a lot more thoroughly across the industry for various applications. I think this is, useful in tox screening. I think it's useful in, maybe cosmetic screening across the board. You could think about environmental toxins. This could be useful for. It is very broadly applicable. And the great thing about it, Ross, is that you don't need to retune it every time you have a new application. And so you can very quickly start to test different types of perturbations, and you can use it in almost any different cell model, advanced organoids, primary cells. We do this in, primary blood mononuclear cells, or PBMCs, which can be derived from patients, and you can do it in simple immortalized cell lines too. So it's applicable across the board.
Yeah, that makes a lot of sense. I'm interested in, where you think VCPI fits into the sort of broader virtual cell ecosystem. So you've got the ARC Institute with the state data set models and the virtual cell challenge. You've got the Chan Zuckerberg initiative. You've got, Tahoe 100 million. What do you think that this work that you're doing with VCPI adds to that ecosystem, or would you differentiate it from the other initiatives that are underway?
Diversity. I mean, every single one of those initiatives that you just discussed are all based on single cell techniques. There's nothing wrong with that, although we do have more and more data showing that there are limits to that technology. But it's really having that orthogonal measurement that's really important. And I think that the complementarity of DrugSeq is going to be important to virtual cells. What's key and what's missing, and this is something that in the future we need to do, is we need to bridge that gap, as I alluded to before. What's the power actually modeling on both these types of data sets? There are some aspects of single cell that I just can't replicate with DrugSeq. I'm not saying it is great for every purpose, but I think for virtual cells, it can really be an accelerant, and it could be really a powerful tool for folks to improve models that maybe have fallen short before in just single cell data. But if you're looking for just pure volumes of data, lots of it zeros, but also actual integers above that, and maybe for heterogeneity too, single cell is the right way to go, and cell classification, et cetera. So I think both methods are actually quite important, and I love to see that come together, and for these initiatives also to bridge and come together too.
Yeah, and that makes sense, that if there's a lot of focus industry wide on single cell data, and there are a lot of sort of methodological problems that are yet to be resolved with how you work with single cell data, and also you've got the cost constraint of trying to generate single cell data, that if there's an opportunity to build on that single cell data or integrate with that single cell data in a way that is more cost efficient, that is reproducible, and you can sort of target the data set at the set of compounds that you want to focus on, then you've got an opportunity to do something special at a very high value. Am I thinking about that right?
That's exactly right.
Yeah, awesome. So another initiative that you all have underway is you work on the Antibody Developability Consortium, this federated learning initiative with Apheris, and people who have listened to multiples of the Data and Biotech podcast might remember Apheris CEO Robin Rome came on and talked about federated co-folding. So I don't want to talk about sort of like what federated learning is, or what Apheris does, but you've got this theoretical fix for data sharing in biotech, where everybody gets the benefit of the model, your data stays private, but everybody gets the benefit of training on each other's data, so that they can collaboratively they can get further than they would otherwise, without giving away the proprietary data that they have internally. So I'm just interested in what your role is in the Antibody Developability Consortium, and what you think the benefits and the limits of that model are.
Yeah, Robin, first off, has been an amazing thought leader in the field of federated learning, and Apheris has been an incredible partner to us. We're excited about the Antibody Developability Consortium because it is very unique in that we are not training on historical data. Okay, and so this is where Ginkgo's role is really special. Most consortia today, in fact, all of them that I know of, with a caveat, we'll go back to that, are based off of historic data. So that is, you have some also in developability that are a number of pharma partners coming together. They have historic data that's been collected over years, if not decades. They put this into a federated, kind of architecture, and then they learn from those data. I actually compare that to doing a puzzle in the dark. Okay, meaning that you can't see the data in that federated environment, but you can train on it. So you're passing the weights back and forth, but you're not actually seeing it itself. You're making a lot of assumptions into how that data was actually generated and the assay parameters and all the other things, right? You can see the distribution of those data, but you don't really have necessarily details. And let's face it, there probably are some limitations to the way you collected that data. In fact, one company alone probably has a change in the methods that they used from the start of that historical data time point, right, to the end of it. And so there's probably diversity even within one partner. And then you're training on this across multiple partners. We'll see how that turns out. I think that there will be limitations to it, but certainly could be helpful. And I think it's important for me to acknowledge the amount of effort and work that has already gone into establishing this type of relationship between multiple pharma partners. That is an incredible amount of progress to get pharma to partner in this way and to actually be pre-competitive and say, let's come together and learn together, right? That is not an inherent trait of pharma, right? Pharma oftentimes is very hyper-competitive, they're very secretive, and they don't work well together. There are exceptions to that, and I think that in the AI field, we are certainly seeing consortia be an exception to the rule. And I think that some of the work done with Amgen and others, through either the Fate Consortium or the Melody Consortium, while there may be limited returns in terms of model performance, certainly just being able to do that activity with multiple farmers involved is impressive. So we've gone through those business development, those partnering kind of challenges as well. It's worth the effort, in our opinion. We're right on the level or right at the stage of taking this to the next level where we're going to be in the data generation phase. And this gets back to like the special nature of our consortium. And that is, instead of using historic data, we are actually building a made-for-purpose training data set that is going to sit in the center of this federated network. It is going to be designed through contributions of our member partners. Currently, we're looking at 3 or 4 founding members. We can go up to 5, maybe 6 or 7. I think there's really no limit, but we're kind of planning this around the idea of 5. We'll have up to 10,000 antibody sequences total. Each partner will actually submit 2,000 sequences, and they will be able to get the data back on those 2,000, the actual data, the actual, kind of CSV file coming back, the data return. On all the other ones, the remaining 8,000, is that they will be able to train on that through this federated environment. And what's neat about it is that this is meant to be very collaborative. And there's a lot of sharing of ideas and best practices, but at the same time, it is reserving each member's autonomy to be able to do it on their own too, meaning that you can bring your own AI ML team to this, and through the federated environment, you can train on that central training data set, right? At the same time, Ginkgo, with the help of our collaborators and our academic partners, we are actually building a central model ourselves. And so that will actually act as a baseline for everybody else. And if their modeling approaches maybe aren't as good as ours, well, at least they can fall back on ours. Or if their modeling approaches are great, well, then they keep their model, and, maybe their model is better than anybody else in the consortium. They don't share that, right? So I think that it kind of balances out those different incentive structures. But what I'm really encouraged by is that we're going to develop all these data with the same exact protocol. that we've developed here at Ginkgo, and that we've been delivering data to our customers for over the last 2 years. And we're going to do this at a scale that I think has never been done before. And we're going to do it on behalf of our partners who are early adopters here and willing to work in this collaborative environment. I think that's a really nice outcome that we're right on the precipice of seeing.
Yeah, and if I understand correctly, if you're able to sort of demonstrate the value of the models that you're producing here, then what it does is it creates a clear path that embeds your the protocols that you've established, the data generation procedures that you've established in the heart of these really valuable models. Should I think about that as like a strategic consideration from Ginkgo's perspective in Datapoints involvement in this, or how would you change that characterization?
Well, I think it's always been a bit scary for me to think that there are certain standards of what we call a drug-like molecule when it comes to an antibody, yet we have different ways to measure those standards, right? As a field, we can all agree that it should have a certain amount of hydrophobicity and polyspecificity and these types of measurements. But if we're actually taking different approaches to measuring those and having different outcomes, that's probably a bit of an exclamation mark on the field in general, right? So I think that the need to standardize these assays and these outcomes are actually emergent and necessary for the industry as a whole, even without AI. But it becomes even more important when you're starting to model and train, et cetera. Now, the caveat that I showed before in talking about, kind of made for purpose training data, and this is, I think, adjacent to it, but it's not on the nose here. And that is, I'm really excited to see the work that Lilly Toon Lab is doing and the way that they've built out their federated learning. Now, that one is not so central, but rather they have their models that are available to, I think, now 100 different smaller biotechs that are part of their network. And the biotechs is a kind of an exchange of services here where the biotechs can access the models that Lilly develops, but in exchange for actually contributing their data to it. Now, the way that they do it is that they have certain multipliers or kind of credits, if you will, and you get a higher credit if you use a Lilly-like protocol, right? And I think that this is a really interesting thing. So I think the field is moving towards having these more established protocols, still leaving room. You don't have to use the Lilly protocol, but you're incentivized to do it, but still leaving room for some divergence from this. And I think what's interesting about the Ginkgo Apheris Consortium is not so much that we're trying to enforce our standard on everybody else, but rather we're trying to actually take everybody else's standard and come up with a central solution that is actually good for the industry and matches a lot of the data that they already have. And that is one of the existing preliminary conditions, I would say, to participating in the consortium, is that we've done these pilot experiments with all of our members. They already know what the coherence is with their existing data. And so I think the good news is that we're already pretty well aligned, and it's all about refinement from there.
Yeah, that makes a lot of sense. And anybody who's interested in Toon Lab, we did have Eliza Apple on the podcast to talk about awesome. Toon Lab on a previous episode as well. I think that if I were to update what I said earlier, it would be that the establishment of mutually beneficial standards encourages everyone to invest in data generation and train more models that can build on each other, and where each data point that gets generated is more valuable to each individual organization and more valuable to the ecosystem at large. And so, as the value of each data point in the market goes up, that's beneficial for data generation generally. And so that's
absolutely
that makes a lot of sense. You've built this autonomous lab designed for machine to machine, or you're building this autonomous lab designed for machine to machine operation, and you've got this collaboration with OpenAI. Can you talk a little bit about the work you're doing? And I'm really interested in what a lab in the loop architecture looks like.
Yeah, this is really neat. You can come by our Seaport facility. This did not necessarily exist this level when Datapoints started. We have a lot of different automation throughout Ginkgo. One piece of automation that we're investing heavily in, though, is a unit called a rack, a reconfigurable automation cart. We've actually been developing this internally for many years now. It was actually started at Zymergen, and we acquired Zymergen. So it has about 7 or 8 years of product development to it. And it really finally reached the point now, and I think that the field is actually ready for it, in that we're establishing these large system like autonomous labs that are able to generate scientific data, not just at high throughput. We've seen that before with work cells. That's some of the technology that Datapoints uses from other kind of automation providers. That's not so new. What's new about the autonomous lab is that we have 100 different instruments on one system, and it's really meant for diverse data generation, right? And so it can do more than one protocol at a time, 80, 100 protocols at a time. And it really is replicating what is happening in the lab today from a physical architecture perspective. That is, if you go into a lab, you see this linear layout where you have these black bench tops with instruments on each, on each section of it. And then you have a human that actually goes about and transfers samples from instrument to instrument in the order that they need it and at the time points that they need to do it. This replicates that. It is a linear setup. However, it is a magnetic rail that transfers the plates from one instrument to the next. And instead of a human dictating when that should happen, it's, of course, a human writing the protocol. But actually, you have then the Catalyst software that we have driving where the plates need to go at a specific time. And it's orchestrating all these in real time so you can have multiple protocols at once. Now, I mentioned, and I said, of course, it's a human designing the protocol, but that's not always true. And so we did a really neat collaboration with OpenAI where they used GPT-5 to actually design experiments to optimize the output of a cell-free protein expression system. And so this is really important. Cell-free protein expression systems, simply speaking, are you take away the cell membranes, you have the rudimentary mix of what you need in order to translate proteins and make proteins. And you're able to do this with a number of components, but those components need to be optimized. The ratios of those components, which ones you add, are there any that you can remove? This is a big multi-parameter problem. And of course, there are consequences to what your recipe is. That is, you can not only have higher or lower titers, but there's also a cost perspective. And so the goal for the industry, and cell-free protein expression is used in some drug development and actually commercially approved drugs. It's also used in other industrial applications. But the idea is that you get a higher titer of protein expression from each volume of lysate that you use. And then you have a lower cost point. That's the goal, right? So more protein, less cost. We did about 36,000, 40,000 different reaction conditions using GPT-5 to actually select those reaction conditions. What was amazing about this is that GPT-5 was writing its own ELN. And so we often talk about what is the interpretability of AI models. In this case, you actually had the LLM telling us exactly why it chose to do what it did. And it also was looking for new reagents. Like, what are some reagents that we haven't thought about before? And they actually suggested some pretty unique ones. Some of them we couldn't actually source. There is a human component to actually doing procurement for the reagents and loading the reagents onto the system. But for a large part of this, GPT-5 was actually designing the experiments and the protocols, and then the autonomous lab that we have, what we call Nebula, based on these rack setups, reconfigurable automation carts, they were really doing the experimentation. And I think this is a really neat example of what can be done with autonomous labs. Now, it's not just a GPT-5 that can do this. The humans at Ginkgo do this. We also have a cloud lab interface where other scientists can actually access this and submit samples as well. And there's even some sense that maybe you could have a bake-off one day, almost like the Go type of competition, where our cell-free protein expression did reach a new goal. We successfully had the lowest price point per titer in the industry that has ever been reported before, and GPT-5 did that. There are some academics here, in fact, one that probably was influential in the way that GPT-5 learned what some of the best experiments could be. Michael Jewett from Stanford, who is an expert in the space. Maybe one day we could actually see him, and you actually have a human that's designing experiments. And if he had the horsepower of Nebula behind him, perhaps he could even come up with a better outcome. So I think this is a really interesting way to think about where the field of autonomous lab is going, and the role of humans in that loop.
No, I mean, that's an entire podcast episode to itself. But as we head toward the end here, I just have a couple of final questions for you. So, like, if you were advising, a mid-stage biotech on how to think about their data strategy in the context of the bio AI landscape, what would you tell them?
I think it depends on who the tech company would be. Mid-stage biotech, are you looking to make drugs, or are you actually looking to monetize a model? If you're looking to make drugs, I would advise you to stay on track, think about the critical path, but be as hungry as possible for data on that critical path. Very similar to the ADME story that we talked about before. Make as much measurements on your early series as possible, and it's going to benefit you down the road. I would say for the modelers, you have to think about your platform as a hamburger in many ways. That is, if you're going to build a hamburger shop, you're probably not going to grind your own meat, bake your own bread, kind of grow your own lettuce. You're going to source a lot of these things. But what the really important aspect is understand what is your special sauce. What are the 1 or 2 ingredients that you need to own that makes your hamburger special, and really invest in your data there and make sure that it matches your model architecture and is consistent with your entire business, strategy. But once you have that, I would say that you really need to start investing in a way. And again, it's not just quantity of data, it's the quality and diversity of the data that really matters.
Yeah, no, as you're talking, it sort of strikes me that, like all of the success in the protein modeling space is sort of based on the quality of the data that was available and the inductive bias that the modeling architectures had available. And so this, beautiful coupling of data and model is what led to the success that we've seen from AlphaFold. And so, as a modeling company, that's what you're searching for, is that sort of synergy between the data that you're generating and the type of model that you're architecting. Do you think
that's absolutely right? That's right. And also the ecosystem tools that you pull in to help you, to give you that uplift as well, is that, you don't need to invent everything from scratch.
Yeah, that makes sense. Do you have any final thoughts that you would want to leave us with as we prepare to let you go?
Yeah, this has been a really fun conversation. I want to end it on a high note. I've expressed a number of criticism. I've talked about gaps and maybe offered some solutions on this. But I've never been more excited about where we are in the space. I consider this to be a point in time, and I recognize it's an early point in time. As scientists, we are always a little bit more cautious. We're data driven, and once we see the data, then we can make informed decisions and move forward. And I think what we're looking at now, and we talked about scaling laws and what type of scaling works, what doesn't. That's a barrier, but we're going to solve it, right? We talked about how do you actually understand what the best models are, and how do you empirically evaluate this across the industry. Again, that's a data problem. We're going to solve it. All of these things are solvable, and the good thing is that us as scientists, especially in the biotech world, we are very well equipped to actually solve these problems that are currently presenting themselves. I think it's just a matter of time and, of course, investment. But the investment is going to continue to increase as we start to see what the return on the investment is. And I think that's already showing up and will continue to do so in the years to come.
Yeah, that's great. Great place to end it on. Thank you, John.
Thanks, Ross.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.